Welcome to the Cell Membrane Structure, Permeability, and Transport Animation Project by Cole Simmons, Iain Jacob, Yang, and Ethan Satota. My apologies for any speech mistakes I make as I go along. There is quite a script here. I will begin with the topic of the structure of phospholipids. Phospholipids contain a hydrophilic head and a hydrophobic tail. A long hydrocarbon chain makes it hydrophobic. Phospholipids will orient themselves with their tails facing each other and the heads facing outside into the aqueous external environment and the internal environment. This is because the heads are hydrophilic and the tails are hydrophobic. Embedded proteins serve as a channel for specific molecules to pass through the cell membrane that normally would not be able to. Surface proteins serve as a signaling function between cells. These proteins are able to stick to the plasma membrane due to the polar interactions with the hydrophobic heads. Membrane proteins will usually have a hydrophilic section in order to be able to stick to the membrane's hydrophilic heads through polar interactions. Some proteins, such as embedded proteins, will have areas that are hydrophobic. The plasma membrane is a fluid mosaic because it incorporates several molecules such as embedded proteins, cholesterol, and phospholipids to make up the membrane, which in turn make certain processes simpler. For example, embedded proteins help with the transport of molecules, glycoproteins, and glycolipids, and can act as identification tags to create cell-cell bindings. Cholesterol acts as a fluid buffer by resisting changes in membrane fluidity due to temperature changes. The membrane structure of biological membranes influences selective permeability by having the ability to regulate transport across cellular boundaries, a function that is essential to a cell's existence. For example, the lipid bilayer allow the hydrophobic membrane of the interior to pass through. Proteins built into the membrane play key roles in regulating transport. Hydrophobic substances are soluble in lipids and pass through membranes rapidly. On the other hand, polar molecules and ions cannot pass freely across the membrane themselves. Specific ions and a variety of polar molecules are able to move across the membrane and avoid contact with the lipid bilayer by passing through transport proteins that span the membrane. For example, transport proteins called channel proteins function by having a hydrophilic channel that certain molecules of atomic ions use as a channel to pass through the membrane. Water is able to cross the membrane with a channel protein called aquaporins. Aquaporin allows up to 3 billion water molecules per second. The cell wall is important in helping maintain the cell's water balance when immersed into a hypotonic solution, meaning water enters the cell faster than it leaves. For example, in a plant cell, the cell wall will expand only so much before it exerts back pressure on the cell that opposes further water intake, which is called turgor pressure. However, a cell wall is not beneficial in a hypertonic environment where the cell is losing water. Passive transport is essentially when a substance moves from an area of high concentration to an area of low concentration. This phenomenon is called diffusion and requires no energy from a cell. In water, when the concentration of a solute is higher and when there is an equal volume of water on two sides, the water will go on that side, which is called osmosis. This is because when water molecules cluster on polar molecules, the concentration of water molecules decreases. Most cells will incorporate transport proteins into the membrane, which simplifies the transport of ions and polar molecules as movement is normally impeded by the phospholipid bilayer. Active transport when a cell expends energy in order to move a solute across the membrane. Most of the time, ATP will be used to supply to requirements for active transport. A specific case of active transport can be seen in the use of sodium-potassium pumps. In animal cells, the concentration of potassium ions is significantly higher than the concentration of sodium ions. Thus, in order to bring the potassium ions into the cell will pump out 3 sodium ions in order to bring in 2 potassium ions while expending ATP to change the shape of the sodium-potassium pump. Large molecules such as proteins and polysaccharides cross the membrane in bulk package in vesicles. Live active transport, these molecules require ATP energy. And here is the diagram displaying this this very process. Thank you for watching.